How can we measure the potential transmission of a disease? How can we combine genetic and prevalence data to measure the transmissibility of a disease? Can we use computationally efficient methods to estimate the reproduction number from multiple data sources? We explore these questions and more in this episode of SBIDER Presents.
This is SBIDER Presents, a series produced by the SBIDER Podcast Hub. My name is Laura Guzman.
And mine is Ed Hill.
In this episode, we are joined by Alicia Gill, a PhD student from the Warwick Mathematics and Statistics Doctoral Training Programme at the University of Warwick. We will be discussing work done as part of Alicia's PhD project, investigating how to estimate the effective reproduction number, incorporating genetics into epidemic modeling, supervised by the SBIDER members Xavier Didelot and Richard Everett and Jere Koskela. We will be discussing the research article, Bayesian inference of reproduction number from epidemiological and genetic data using particle MCMC, available as an open access archive from November 2023.
Hi Alicia, thank you very much for joining us on this episode of SBIDER Presents.
Hi.
Welcome to SBIDER Presents.
Thank you. Thank you for having me.
To begin, would you first like to share with the listeners how you gained a general interest in the research areas of epidemiology, genomics, and mathematical modelling?
Yeah, okay. So I have always been interested in these things separately. So I've always been interested in maths and I've always been interested in biology. So at A-level, I did maths, further maths, and biology. And before I wanted to be a mathematician, as a kid, I wanted to be a doctor. I was always interested in these things as separate entities. And then on my master's, I learned that you can combine these things together. So you can do mathematical modelling in epidemics, and you can learn things about the epidemics and learn things about maths at the same time. And you can do the same thing with the genetics. So I did all sorts of modules like population genetics and medical statistics, and it really, combined two things that I'm interested in into one subject.
Yeah, that sounds a perfect field for this. So your PhD project covers very relevant subjects. First, inferring the reproduction number of an infectious disease, and second, mixing different sources of data, including genomics. What was your motivation to work on these topics?
So, I'm sure this is the answer that everyone gives, but the pandemic. The pandemic happened, and so we know from that how important mathematics is in these real-world applications for epidemiology, for inferring the reproduction number. With the genomics specifically, COVID had way more sequencing done than any other epidemic done previously. I think it's something like 17 million COVID sequences were taken over the course of the COVID pandemic. And there are not very many methods that can really harness that amount of genomic data to tell us things about infectious disease epidemiology. So that's why I want to incorporate the genomics.
Yeah, it really has been a different order of magnitude in terms of the amount of data that's being collected, or that type of data that's being collected for this particular pathogen. So it's interesting looking forward as to what could potentially be done for other pathogens and other systems. So with this particular study, so Bayesian inference of reproduction number from epidemiological and genetic data using particle MCMC, what were the main goals of the study and what were the knowledge gaps at the time that you were hoping or envisaging the study helping to address?
Okay, so I will start with knowledge gaps. So there are lots of methods that can take some kind of time series epidemiological data, so case count data or incident data, that kind of thing over time, to infer the reproduction number trajectory. There are also a few methods that can take genomic data, so you can take a tree, like a phylogenetic tree, which shows you the evolutionary history of your pathogen, and you can infer the reproduction number from that tree. But there are very few methods that can combine both sources of data together explicitly, and so that was the aim, to see if combining the inference of these things into one model improves inference compared to looking at epidemic data and then looking at genetic data and seeing if they corroborate or not.
Oh, so how did you approach that problem? And were there methods existing already, or you developed your own?
We wanted to take a Bayesian approach to the problem, because Bayesian statistics is a really natural way of incorporating multiple sources of data together. We had to construct our own model, because it is not obvious exactly how a tree relates to an epidemic. So we needed to figure out how we wanted to model that relationship. But once we'd done that, the statistical techniques already existed. So we use particle MCMC. Particle MCMC is like normal MCMC, but instead of using the true likelihood, you have to use an estimator of the likelihood. And we can construct this estimator by essentially simulating a bunch of possible epidemic trajectories and a bunch of possible reproduction number trajectories, and then averaging over these samples to get the likelihood that we use inside of the MCMC. So instead of trying to explore the whole space directly, we sample many, many trajectories from the space and use those samples to decide which bits of the space have high likelihood and which ones don't.
So from this approach where you're identifying what are the more likely sets of parameters given the data that we have available.
Mm-hmm. Yes, exactly.
That's great. And so from this quite new model that you developed and then with the statistical techniques applied, what are then the main results that you found from your study? And were there any findings that were unexpected or surprising?
The main thing is that it works, which is great. So when you combine two sources of your data together, we do find that we have more credibility in our results. So we have more confidence that the results that we're getting are correct, and the credible bounds around our results are tighter. So we get more robust results when we combine multiple sources of data together compared to just looking at one source of data or the other. In terms of surprising things, you don't actually need very much genomic data for this to be true. You can have a tree that has like 10 or 20 sequences in it, and that still gives you a lot of value. So we don't even need to be talking about 17 million sequences. You can collect very little genomic data, and it will still really help you with your inference of your reproduction number trajectory. And the other thing that I found surprising is that one of the parameters we have in our model is the reporting probability. So what proportion of our cases have we observed? Have we observed a large amount of a small epidemic or a small amount of a large one? And introducing this genomic data really helps to pin down this reporting probability parameter, even though this is slightly counterintuitive because the reporting probability is a purely epidemiological parameter, not a genomic parameter. But knowing the genomic data helps you pin down everything else enough to give you a really good idea of your reporting probability.
That seems very, very powerful. It's like helping with this identifiability type problem in terms of adding in a different data stream can then help reveal, say, the value of these other parameters in systems such as the reporting rates. And then the fact that you need perhaps just a small number of samples, that seems very useful if you're in a region where not much sampling can be done. But if you just have a few samples, if that can then help with the reproduction number estimates being made and their robustness, that seems fantastic.
So from your perspective, what are the main implications of these findings? I guess there are limitations to bear in mind, or does it give you direction for future work?
Okay, so the main implications, I would say, is that it is worth investing time and money collecting multiple different streams of data. So rather than putting all of your money into testing to find more cases, you might be better off spending some of that money sampling some of the genomics for the cases that you do have. I think it looks promising for other sources of new data, so things like wastewater data. I think there's promise that combining multiple sources of data together into your models explicitly is going to have tremendous benefit, and so that is worth doing. In terms of limitations, so because we want methods that are scalable for the amount of genomic data that we have, I take one phylogenetic tree and I treat that as my data. But actually there can be tremendous uncertainty around constructing this phylogenetic tree, which is currently not being incorporated into the model. So there is some missing uncertainty that is not being propagated through. Whether this is an issue depends on how well resolved you feel that your tree is. And actually, I think for a lot of something like COVID, I think the tree is quite well resolved because we've been building it in real time and we know which things have turned up when and where they've descended from as they've been happening. We're not necessarily taking sequences at the present day and building a tree backward in time. But this could be a limitation to the approach. And I would say the last limitation to the approach is that it's currently using quite simple epidemic models. But epidemic models can be very complex and very bespoke, so I don't take into account an exposure period, or I don't take into account vaccination status or age stratification or things that are quite traditionally accepted in epidemic modelling. My model doesn't do yet. So that would be scope for future work to how to combine state-of-the-art epidemic modelling with genomic data, not just any epidemic modelling with genomic data.
Thank you for outlining those potential directions. But it seems you have a platform from which you can now go from to start exploring these different directions, which I think that's very exciting. And at a bigger picture, or taking a high-level bigger picture perspective, what, in your view, might be the key challenges for incorporating genomics into epidemiological modelling, that's generally as a kind of integrated type field?
So I alluded to this in the answer for my last question. I think scalability is going to be the big issue. Because at the moment, all of the methods that do this kind of thing take sequences and build a phylogenetic tree, this is not going to be scalable for more than a few thousand samples, really, because the methods that we have can do that are things like BEAST, which is an MCMC over tree space, and tree space is like double exponential in the number of leaves. So the more samples you have, the way more bigger the space of trees that you're trying to explore is to find the correct one for then using in this kind of inference. So I think that is a really key challenge, and I think the direction for that kind of thing is going to be figuring out how to include genomic data in a way that bypasses the need to construct a phylogenetic tree. So can we take this sequencing information directly and use that as it is without having to build the tree to then do the inference? Because this building the tree step does not scale very well with massive amounts of data.
So the 17 million burden.
Yeah, that's just not going to happen. 17 million in a tree.
Cool. Thank you again, Alicia, for sharing your insights on your work and the kind of future perspectives on where things might go. I think that's really a really interesting outlook, and I'm excited to see how this work develops.
Yeah, same. Very excited about your work. So thank you for joining us in this episode of SBIDER Presents.
Thank you again to Alicia for joining us today, and thank you all for listening to this episode of SBIDER Presents.